Hi, my name is Brittney-Hien, and I am presenting my poster over morphine withdrawal-induced immunosuppression, which modulates DNA methylation in IEC18 cells. This is my poster, and I'll be going through each section in this PowerPoint. The objective of the study was to show morphine withdrawal and its effect on rat intraepithelial cells, DNA methylation, and immune suppression. So morphine withdrawal can cause immunosuppression. This is seen in Campbell, this study right here, and it showed decreased lymphocytes, macrophages, and cytokines. Morphine withdrawal can alter DNA methylation, and this was also seen as in a study. And so DNA methylation is a heritable phenomenon which increases expressivity of a gene but doesn't alter it. So what we're saying is that morphine increases methylation of genes, and then we were trying to study and see which genes they are and what's going on because of this hypermethylation. Substrates to measure these changes: MECP2, which is a protein that binds to methylated CpG regions of DNA. An increase of MECP2 will indicate an upregulated immune response because of its association with NF-kappa beta. And then IEC18 cells were used because we were also interested in its effect on inflammatory bowel disease. Methods. Okay, so row A was our control, placebo withdrawn. There was no morphine used in that. Row B, we used our morphine withdrawal. So we did 3 days of treatment for morphine and then 1 day where the cells were cleaned and no morphine was given. And that is to stimulate our withdrawal phase. And then cells were harvested and used. Column 1 was our control. Column 2, we used LPS to stimulate inflammation, and that was kind of how we tested if there was an immunosuppression. And then in column 3, we used azacitidine and LPS, and azacitidine is a drug that is used in inflammatory bowel disease, and it's been known to suppress DNA methylation. So we're just interested in that too. So total RNA was extracted from each well, and total DNA was extracted as well. The RNA was converted to cDNA to make it more stable to study. We also did a bisulfate conversion. And so what that is a way for us to see where the methylation is in the DNA. So you can see on the left on this image, the methylated version, which are tagged with these yellow dots. They are methylated on these Cs, and through the conversion, they remain Cs, and through PCR, they remain Cs. While in the unmethylated version, there's no methylation. So through the conversions, they become Us, and then through PCR, they are becoming Ts, because that's just how it is in PCR. The primers used for this, so the cDNA, which is our RNA, we use MECP2, which, like I said, binds to hypermethylated sites on the DNA. And then we use MORE, which is a mucoid receptor. And then GLS is a region of DNA that when it is transcribed, it becomes able to convert glutamine to glutamate. And that's a neuroexcitatory neuron. And we were using that to see if inflammatory bowel disease causes an increase of this, which would indicate more synapses for pain stimulations. And then DNA, the primers we used was GLS, MECP2, and MORE. The results. So this is the table showing our cDNA results. And so morphine withdrawal, which is on this right side, and then induced with inflammatory products, which is LPS, we can see a decrease in MECP2 and GLS compared to the control and compared to no morphine at all, which indicates this was an immunosuppressive state. And this one is the DNA methylation analysis. And so we are seeing how hypermethylated these genes were. So the higher the band, the more methylation. And so in the morphine withdrawal cells, which is on the right again, induced, there was an induced gene expression. So there's increased gene expression in MECP2 and more. And so that's just saying that more in MECP2, because of morphine withdrawal, they were hypermethylated. And so our conclusions: morphine withdrawal-induced immunosuppression altered the methylation of GLS more in MECP2. Results need to be repeated because we were on a short time frame. And so we need to repeat these to make it more statistically significant. And then our future studies will be more on how exactly these genes make proteins and how this downstream effect occurs. And then just in general, morphine withdrawal immunosuppression in its relation to DNA methylation is still a very under-researched area in epigenetics. And yeah, here are my references. And then acknowledgements, thanks to the Mentor Mentee program and Dr. Doss for guiding this research. And then thank you for watching.